Hello, my dear students. This mini lecture will discuss some special properties of enzyme proteins. Be sure you're ready to take notes as you watch. By now, you are familiar with the four types of macromolecules. There are carbohydrates, lipids, proteins, and nucleic acids. Although carbohydrates and lipids stand alone, well, kinda, because technically fatty acid chains are really just carbohydrates with alcohol attached. Proteins and nucleic acids are very closely related. So let's explore that a little bit more. Nucleic acids make DNA. DNA is transcribed to RNA, which is also a nucleic acid. RNA is translated to amino acids, which fold together by covalent bonding to form proteins. So amino acids are the basic building blocks of proteins. There are a bunch of different kinds of proteins, but in Module 2, particularly for this mini lecture, we'll talk about enzymes. In the case of enzymes, amino acids are folded together to make proteins that catalyze reactions. So what do enzymes do? Enzymes are catalysts. In biology, a catalyst is a protein that speeds up a biochemical reaction. So instead of waiting for the right molecules to bump into each other accidentally, which may or may not ever happen, we can think of enzyme catalysts as matchmakers. Enzymes connect with substrates to create a product. For example, salivary enzymes in the mouth and digestive enzymes in the stomach help break down complex carbohydrates in our food to simple carbohydrates, and then the molecular components that our cells need to keep our body running. Enzymes are responsible for breaking large polymers down to small monomers, or connecting monomers or smaller components to create polymers. Sometimes enzymes catalyze a rearrangement of monomers to create a new polymer from the same material. An essential part of protein function has to do with its structure. Have you ever tried to fold a fitted sheet? It's weird. It does not fold the same as a flat sheet. The structure of the fitted sheet affects the way it folds. A protein shape is critical to its function. This is true for all types of proteins, but let's stick with enzymes for now. An enzyme can bind to a specific substrate at an active site. If this active site is altered because of local changes or changes in overall protein structure, the enzyme may be unable to bind the substrate. If the enzyme cannot bind the substrate, then the protein cannot function. When an enzyme successfully binds to a substrate at the active site, the substrate undergoes what's called a conformational change. This is a change in the shape that allows the protein to function. Let's demo this with my lock and key. Let's demo this with a lock and a key. Now, in this instance, we have two substrates, the housing of the lock and the locking pin or the bar, okay? And we have one enzyme, the key. So we have two substrates, which means we need two active sites. Can you guess what the active sites are? Okay, the active site for the lock housing is going to be the keyhole. And the active site for the bar is this little notch here. Okay, now, if we have the right key, without the enzyme, we have our two substrates. They're not working together. They're not doing what they're supposed to do together. The lock has absolutely no functionality. It doesn't do anything. But when we have the right enzyme, so the right key comes along, it fits. Now our lock is functional and it can lock. So we see that the bar is holding itself to the lock housing. So now the lock can function the way locks are supposed to function, which is to keep things out of other things. Okay, so keep things out of a place. Now, each enzyme, just like in this example, each enzyme is specific for the substrates that it interacts with. It can't be any old key. It has to be the key that is cut in the right pattern to fit into this keyhole and activate the locking mechanisms inside here. Without that, the lock won't lock because the bar cannot be clicked in, and you couldn't unlock it. Okay, so this is a checks and balances system that protects our enzymes and protein function from being activated by any old enzyme or any old substrates that happen to bump into each other. If an active site is accidentally exposed, it's okay because we don't have the right enzymes there to activate it. Okay. To understand how the protein gets its final shape or conformation, we need to understand the 4 levels of protein structure: primary, secondary, tertiary, and quaternary. We'll start with the primary structure. The amino acid sequence in a polypeptide chain is the primary structure. This example here on the right shows the amino acid sequence for the insulin hormone. We can see here that there's an alpha chain with one set of amino acids and a beta chain with another set of amino acids. These 2 strands of amino acids are held together by disulfide bonds. We can see these double S links here and here, and then there's another set here. Now, this configuration of these particular strings of amino acids can only make one thing, and that is the hormone insulin. My genetic students will remember that cysteine, the amino acid cysteine, is stuck up. So we can see cysteine here, 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 and here. There's 2 right there. Cysteine only likes to bond with itself. It doesn't bond with anybody else. So the polypeptide chain can take advantage of that property and use the cysteines to form disulfide bonds to link these 2 amino acid chains in just exactly the formation needed to create insulin. Okay, so the first one was primary structure. Now let's look at secondary structure. Secondary structure is determined by local folding of the polypeptide. Local folding means that the particular folding patterns happen in certain regions of the polypeptide chain. Here we see the most common type of local folding with an alpha helix. You can see this balled up helical shape, and a beta pleated sheet, which we can see over here. These folding patterns are created from hydrogen bonds between the alpha and beta chains of the primary structure. So let's look at, if we look at just the pleated sheet, here's one chain of amino acids, here's another, and we can see the hydrogen bonding here that from the same chain that allows it to kind of twist on each other and wrap. And we can see the hydrogen bonding, the way the hydrogen bonding happens here causes this spiral helical shape. Okay, and then there is the tertiary structure. Tertiary structure is 3-dimensional. This structure is created from multiple chemical reactions between R groups or functional groups of each amino acid. Although in secondary structure, hydrogen bonding is the name of the game, in tertiary structure, we follow the basic rules of attraction. Like repels like, and opposites attract. My genetic students will remember that the first rule for tertiary structure protein folding is that the hydrophobic nonpolar amino acids tuck themselves in and make a hydrophobic region in the middle of the protein so that they don't have to interact with water in the surrounding environment. The hydrophilic amino acids assemble on the outside to cover the hydrophobic region. And quaternary structure is the final resolved protein structure with the active site revealed. So if we look here, we can see just to go from the beginning to the end, primary structure, string of amino acids all together. Then we have the secondary structure with the beta pleated sheet and an alpha helix. And those 2, through special bonding, fold together to make the 3D tertiary structure. Where the R groups link together, so the hydrophobic parts would be tucked in there in the middle, and all this exposed to the outside would be hydrophilic. And then this is converted to the quaternary protein structure that has the active site. Each protein has its own unique sequence and shape that chemical interactions hold together. If the protein is subject to changes in temperature, pH, or exposure to chemicals, the protein structure may change, losing its shape without losing its primary sequence in what scientists call denaturation. Denaturation is often reversible because the polypeptide's primary structure is conserved. So although the 3D shape may fall apart, you still have that string of amino acids in the primary structure. So this allows, under the right conditions or when the right conditions are resumed, for the protein to refold into its secondary, tertiary, and quaternary structure and resume its function. On the other hand, sometimes denaturation can be irreversible, leading to loss of functions, especially if those bonds holding the amino acids in the primary structure polypeptide sequence are broken. One visual example of irreversible protein denaturation is a fried egg. So here on the left, we see the albumin protein in the liquid egg, which is also known as egg white. So this part right here, it's very clear. It's jelly. It looks like this. All right, so then when we heat the albumin protein, it turns into this opaque white. So this is the denatured version of albumin protein. It has lost all its function, although delicious. Now, protein folding is critical to its function. Scientists originally thought that the proteins themselves were responsible for folding process, right? So they've just somehow messaged to themselves that they needed to fold. But recently, researchers have discovered that they often receive assistance in folding process from helpers or chaperones. So chaperones associate with the target protein during the folding process. They act by preventing polypeptide aggregation, that may compromise the structure, and they disassociate from the protein once the target protein is folded. So this concludes today's mini lecture. I hope you find it useful for the upcoming enzyme substrate activity. Good luck.